Good evening, everyone. Welcome to the October CCHBA meeting. Just a few notes to let you know that we still have the state fair going on. This is the last week, so if you're wanting to volunteer in the booth, please get with my dad, John at SabineCreekHoney.com, if you still are interested in volunteering to man the booth. It's been a great time so far. We've had lots of people coming through. And I think the groups that have come through have been very interested in what we have through the TBA booth. We've definitely passed out lots of stuff concerning different clubs around the area, and then also CCHBA. I know that there's been people asking about how to join already, so it is getting some good points out to people to know about beekeeping. We'll also be having coming up soon our elections for the Collin County. I'm putting myself back in for presidency. If there's anybody else that wants to volunteer for a position, please be on our board meeting next week. We'll do this on the Monday at 6:30. Let us know, and that way we can start adding you to the list, and then we'll figure out how to do the voting process. We're still on a Zoom meeting, but we'll get that information out for all those positions that are eligible for election, and then who's been nominated, and then that way you can put in your vote. But I am eligible for another year, and I think most of all other group is eligible for another year for our second year. So we'll have that available to you come the November meeting, and then we'll do our voting then, or however we decide to do that. If we do an email vote or something like that, we'll figure it out. All right, so Blake is not with us today, so I'm gonna kind of run over the tips for October, just some things to be thinking about. Thankful for the rain that happened yesterday, and I'm guessing we're supposed to have quite a bit coming the next couple of days. So it's gonna be great for the fall flowers. If you've actually looked out in your fields or around the areas, especially for our area here in southern Collin County, we definitely see the goldenrod, we see aster, the broomweed. So a lot of this in the rural areas is starting to pop up. The Maximilian sunflowers are up also. So there's lots of bee forage out there. I've been seeing the bees for our observation hives have been pulling in lots of pollen. Why we've been having them sitting out there, plus some of the ones in the field. So I've been seeing lots of the yellow pollen coming in. So they're bringing stuff in. So just make sure you're checking on your bees. You want to make sure that you're checking their honey supply. With these fall flower flows, you should be getting a little flow in. So they may not be going to your sugar water if you're feeding them as much as they would be going to the flowers. So, just be advised that you want to have about 35 to 40 pounds, maybe upwards of 50 pounds. So like I said, just do what we call a hive check, going back to the box on top or your super, picking it up, and it should feel pretty heavy. If it feels pretty heavy, or if you looked in there and they've gotten plenty of pollen and honey or syrup in there, then you're doing well. So just be advised that this needs to be checked on. You don't want to be waiting till the last minute and all of a sudden it gets cold on us, because it can be cold at any time. Texas has a weird weather patterns. We could be cold one day and hot the next day. So just be sure that they have plenty of stores. If you have your queen excluders on, I would go ahead and remove those now, unless you're using your supers on top to do some of your honey stores. Then I would definitely, I would leave the queen excluder on, just so you don't mess up your supers. If you have the double deeps, or you've got other boxes on top of that, maybe some mediums that you're going to leave on, then you can, just like I said, just make sure you got the honey stores, and then you remove that queen excluder so that cluster can get really tight. So that's the way you make sure that queen stays warm while she's in there, and they can still lay eggs during this process while it's cooler. So if they've got her nice and warm, so she can stay inside that brood area. As far as screen bottom boards, it's not necessary that you cover them up. I mean, like I said, we don't have any extreme winters except for this last February. That was a freak cold front, but Normally our Texas weather, you can basically what we call put up some wind breaks. You can drop your hives down if you can to about 6 inches off the ground, or like I've told some is if you just build your little barrier, maybe leave like an inch or two around the bottom. If if you put wood up around there just for them having ventilation, but it's not necessary that you have to cover them up. You can cover them up. I would do something where you could slide something in and slide out pretty easy. That way, if we do get where it's a warm week or a few days that it's getting at in the warmer temperatures, you can kind of gauge how much you want to add ventilation and just to kind of keep them aired out. So that would be one thing just to kind of watch based off the weather. Like I said, we don't have normal Texas weather. If you have weak hives, that now's the time to start really looking at them to see if you want to combine them with a stronger hive, or if you've got a really strong hive that you can move brood. It may have like 6 or 7 frames of brood. You can go ahead and move 1 or 2 with some nurse bees there. You can move those over to the weaker hives and help strengthen those hives. But I would definitely start looking at the weaker hives, and if you're getting down to 3 frames of bees, I would look at going ahead and consolidating with the stronger hive, killing the weaker queen. And that way you can kind of keep your bees surviving with a stronger colony. Definitely keep testing for varroa mites. You want to get those counts as low as possible. Like I said, the closer to 0, the better. But you want to keep doing that. Like I said, you can use the alcohol wash or powdered sugar roll. If you have any questions, like you want to know what kind of treatments to use, you can use the Honeybee Health Coalition.com, and that will help you. You can put in the question, ask you a bunch of questions, and you can answer them, and it'll tell you, what types of treatment you can use, and ask you specific questions for that, and that way you can treat based off of how you answer the questions. But it's a very good tool to use. If you're storing your supers, make sure you're using the paradichlorobenzene, which is the moth ice crystals, not the moth balls, but moth ice crystals. They do sell some other stuff, like a Texas A&M, I think it's called Paramoth. That's another one that you can use. That I think is the same standard, but maybe have a little difference in there. But that can be used in that one. Like I said, just make sure you're storing those. I did see one that some guy posted that he doesn't use anything. I kind of thought it was interesting, but he stacked them with the opening underneath the bottom, kind of like a pyramid, but they had a space under each one. And he said he never had any problems with any wax moth getting up there because there was airflow in between and the sun could get in there. So that's something I thought was pretty interesting. I don't know if I would do it with mine, but like I said, that's something to look at and maybe talk with him. I think he was on the Dallas Beekeepers site. But that was definitely interesting seeing that he just had them stacked up against a fence with the air gap in between them. It was pretty neat. This is also a time during the winter time that if you need to do any repairs to your colonies as far as the woodenware, or start buying new stuff if you're going to expand. If you don't have it already, start working it now when you're not in your bees as much, and that way you're prepared for the spring, especially if you're looking at doing splits or buying new colonies such as nooks and stuff. If you're looking at nooks, and definitely start getting on the list of those peoples that will be selling nooks, getting with them and finding out when you need to order with them. I know for us here at Sabine Creek, we don't start taking orders till January, and we'll have them delivered to you by the end of April. It just takes us a little time, but I know there's other places out there that have them available around March. But like I said, you need to get on those lists if you're going to be doing that, and check with them. Same thing with your queens. If you're going to be looking at getting queens, I know that early on in the March timeframe is when most of the big commercial breeders are looking at the commercial beekeepers and getting their big orders in, and then you can get on their list later on to get queens. Usually it's about April or May to get your queens, but if you, like I said, you can get on the list, maybe they could squeeze you in between. but definitely look around. If you're looking for some of the other areas that still have queens going on now, it's kind of late to be putting queens in, but if you had an emergency situation, I know Kona queens, Big Island queens are some of the ones that they do pretty much your queens all year round. They're a nice warm environment. There's a couple in California, Oliveras queens, and I think there's one called Bee Buzz, but they're up in those areas where it's still warm and they can keep their queens going for quite a while. But definitely want to get on those. Like I said, you should be seeing a bunch of it, of the flowers going on. Like I said, you want to make sure that they're getting lots of their food and be watching this, because I said once our first frost hits, then pretty much everything will start dying off. And like I said, you don't want to be behind when it comes to keeping your bees alive that way and making sure they have the nutrition. As far as pollen patties go, I'm not ready to put any on right now. There are some people that are using some of them just to kind of fatten them up. And I think with the current pollen coming in and the nice little flow that we're getting off the fall flowers, I think it'd be enough. But just like I said, just pay attention. A summer patty can work just as well as a winter patty. The winter patty just will have more sugar in it. I kind of use the winter patties when we're going there. Just like I said, sometimes they can't leave the hive, and you want to have them that carbohydrate and the protein together. Well, for like me, I do external feeding, but if you do an internal feeding where you're putting the syrup in there, I'd probably look at a summer patty and just looking at the protein level for your bees there, and not worry about the sugar content in there. So that is all I have right now as far as the bee talk. If you have any questions, like I said, it's on the Texas Bee Supply site. They have their bee talk where he does most of his bee talk and has tips for the month. You can also look on our calendars. We have our calendar available from this year that we gave out to our members, and that way you can get those bee tips. And like I said, you got the Honeybee Health Coalition to help you with. They also talk about nutrition, but they also have for the varroa mite, doing the different washes and checking how you treat your bees. So I'd like to introduce you to Dr. Rangel. She's been a long time bee lab person down there. She's been running that program for a long time. I know that when I saw it built, she was down there. She's authored many papers. I mean, I can keep going on and on about her. She's done a tremendous job for not only Texas Beekeepers, but even to our local groups with their talks. I know we just awarded her last year the John Thomas Award, and that was a great opportunity to go down there and present that to her and kind of freak her out at the same time. So, but she has been a great asset. She's done some work with queens. I know she's done work with the Africanized bees. So like I said, she has been a real great help for the Texas Beekeepers and has lots of information out there. So Dr. Rangel, the floor is yours, and I can't wait to hear what you've got for us today.
Thanks, Skip, for the invitation. Can you hear me okay?
Yes, we can.
Okay. So first of all, I would like to thank all of you for being here tonight and for this presentation, and also, of course, to the Collin County Beekeepers organizers for inviting me to present. Again, thanks again for the invitation. My new email address is up on the screen because they moved our IT services into AgriLife, Texas AgriLife services. And so now we have this ag before the tamu.edu. I still receive the emails when you just type in jrangel@tamu.edu, but I don't know how long that will last. And then we'll continue using just the ag email address, by the way. A couple of selfish pitches. I am part of a really cool group of researchers that came together during the thick of the COVID-19 pandemic mandates of staying at home, and we created what it used to be called Stay at Home Beekeeping Seminar. That's for a way to teach all kinds of beekeeping classes via Zoom. It was probably the first one of its kind in the U.S. But now that people are free to move around, we kept the program. It's called simply At-Home Beekeeping Webinar Series because it was so successful and people really enjoyed the talks. And you can basically see really top-notch researchers that you would normally see at state beekeeper meetings or at national meetings, that you can actually see their talks online via Zoom. So we've basically been doing this for over a year and a half now, and we have one speaker every month. Last week or a couple of weeks ago, we had Juliana Rangel from the Auburn University. On October 26th, we have Dr. Keith Delaplane from the University of Georgia. And on November 30th, the last one of the year, is actually my graduate student, Alex Payne, who's graduating a few months after that. She does work on the intercross of honeybee viruses and nutrition, very interesting work. And then we'll continue working with more speakers. So this type of flyer looks similar every time, except we keep updating who is presenting. And so it's the last Tuesday of the month at 6:30 p.m. Central Time. You can simply, the easiest way for you to look this up is just type in At-Home Beekeeping Webinar, and it'll show something like this, so you'll recognize it. It's put together by Auburn University, and the PI there is Jeff Williams, but it's really put together more by the Lawrence County Extension Service in that works with Auburn in Alabama. So it's not, so you will see this person, Allison Shable, and she's one of the coordinators of the Lawrence County Extension Service, and she is the one that organizes everything. And so typically we have one of us who's not speaking as a moderator for the webinar. And then if you did not watch it live, then you can watch it pre-recorded for a couple of weeks up on Facebook. And again, it's wonderful because it's free, so anyone from anywhere can join. And the second and last pitch is about our ongoing fundraising campaign for the Nevin Weaver Honeybee Excellence Fund. Thanks to two very, very generous donations, one of which is from an anonymous donor, each of these $25,000, we are getting much closer to our goal of raising $100,000 to the Nevin Weaver Endowment Fund. Most of the work, or most of the funds that we are able to use each month or each year, which is only 4% of the annual interest of whatever's in the corpus, we will be using for graduate research. So your donations are incredibly important to keep up with the rising costs of graduate education and research. The easiest way for you as a beekeeper to donate, I'm not sure if the Collin County Beekeepers has made a donation as a group. If you have, thank you very much. Sometimes I don't know who's made donations, because you can donate either through the A&M Foundation, and you can just Google A&M Foundation and then put Nevin Weaver, and that will take you to a secure site to donate. But the one that beekeepers like to use is the Texas Honeybee Education Association, or the THBEA, through the TBA website. And so you can just go through the TBA website and you'll see something similar to this. And you click and you can donate as little as $1 up to however much you want to do. And then periodically the TBA gives us a bigger check with all the smaller donations, and they are deposited into our account. So thank you in advance. Okay, so I was asked to speak about the ecology of Africanized honeybees in the Americas and in Texas. And of course, I was preaching to a choir because all of you are beekeepers, so you know that the the media have often misrepresented honeybees, and in particular what they call the killer bee, the Africanized honeybee, with sensationalized movies, media pieces, etc., including some of these that you see on your screen that are not actually bees. So you, because you are knowledgeable beekeepers, you would see that the cover for this movie, Killer Bee, is actually not a bee. The same with bees. And so there's a lot of misconception about what the killer bee actually is. And hopefully by the end of this presentation, if you didn't know a lot of information about them, you will. These ones also are not honeybees. So one of the most striking aspects of honeybee biology is their ability to mold to their environment and the variability within and between the races of Apis mellifera, with so many differences in color, size, tongue length, defensive behavior, nesting biology, the dialects of the dance language or the waggle dance, and other behaviors, their susceptibility to disease, and the frequency with which they swarm. So much so that they've been placed in phylogenetic trees that have grouped them into four generalized groups, called the A, M, C, and O groups. The A group stands for African. Those bees are tropically adapted. The M group stands for Northern Europe, and they're more winter adapted. The C group stands for Southern Europe and Eastern Europe, and the O lineage stands for the Middle Eastern group. And so there's, depending on the book you read, there's about 27 subspecies of Apis mellifera recognized by the scientific community. And each one falls into one of those four groups. So this variability in behavior, biology, location is not surprising, because these subspecies have evolved in response to those diverse ecological conditions that go all the way from the very tropical regions to the very temperate regions in Africa, all the way up to Eurasia. So for centuries, we as beekeepers have taken advantage of this variability to select for economically desirable traits. And many of these characteristics are also expressed in their phenotype, or the way they look, that are better adapted to seasonal factors like climate, resource abundance, and our beekeeping practices. So I'll tell you a little bit about the history of beekeeping with Africanized honeybees in the Americas before they got all the way here to Texas. So this whole story starts in 1956 and 1957. You probably have heard some version of the story. In southern Brazil, European races that they were using, most particularly Ligustica and Mellifera, were not surviving well in the tropical region of Sao Paulo in Brazil. They had poor honey production, and they were having issues with sustainability of nests year after year. So Dr. Warwick Kerr was hired by the Brazilian government to breed a more sustainable bee. And so he went to South Africa, and he went to several countries and looked for several lineages or subspecies of Apis mellifera in Southern Africa. But unfortunately, because of the problems with customs in the Brazilian side of the equation, a lot of, or most of the queens that he imported of different subspecies perished while they were being held in customs for quarantining issues, and because the permits were not accepted. And so at the end, they only were left with 35 Apis mellifera scutellata queens that were introduced into Brazilian colonies with queen excluder material, so that they were very well contained within the experimental colonies. But the story goes that the queen excluders from some of those colonies were removed by a local beekeeper, and thus a feral population of a hybrid between European bees and those queens from Apis mellifera scutellata descent was created in Brazil, and thus the Africanized bees were born. That's Dr. Kerr, with colonies of not Africanized bees, but stingless honeybees, which are in the same family, but a different tribe, the Meliponini. So it took about 25 years for Africanized honeybees to migrate north from Brazil to Colombia and Ecuador. So the first introduction was here in Rio Claro in Sao Paulo, Brazil, and they kind of radially started moving. And because of issues with, border controls and everything, it took a little longer for the adjacent countries to receive these hybrids. But anyway, they made it all the way to Colombia, for instance, in Ecuador in 1983. They were moving more or less 300 to 500 kilometers per year, with densities of anywhere from only 6 colonies per kilometer square to up to 108 colonies. So in many regions, there were very high densities. So not very terribly different from the density of European honeybees in some suitable climates for them, like in Europe, their native range. And these feral populations of this hybrid were wildly successful in just integrating into their new environment. So as you can see, they kept moving north into Central America and Northern Central America. And so the first appearance in Mexico was in 1985. And as soon as they arrived into Mexico, in southern Mexico, they immediately, the USDA, Agricultural Research Service, started placing trap lines in the border between Texas and Mexico. So they placed a lot of swarm traps along the line where you have the Mexico border, and here you have the Texas border. And the first Africanized honeybee found in 1990 in the US was in the city of Hidalgo, right there on the border. So this kind of shows what type of trapping was being done. At that time, there were a lot of different trappings. This one was the one done by Texas A&M University, and that's the one that we actually used for the study that I'll talk about shortly. So many of you may have seen a graph like this. When the Africanized bee finally made it into the United States, they created a quarantine because, people were afraid of aggressive encounters with honeybees. So people in every county in the southern US that detected bees that were really hot would send them up for testing. A lot of them ended up at the apiary inspection services. So here, College Station, in Bryan, we received, hundreds of samples from swarms that people were collecting of really aggressive bees, and they were being analyzed back then, as you can see by year, the year here, 1990 and then 91, and started moving northward. The identification was being done by eye under the microscope, looking at physical attributes or morphometrics of the bees. So Africanized honeybees tended to be slightly lighter in weight, and they had a wing venation pattern that was different or unique from European honeybees, and that's how they were being analyzed back then. So you can see this was updated in 2009, and only 2 years later, most of the counties in Texas had been occupied by Africanized bees, and after 2011, they decided, at least here in Texas, that it was kind of assumed that there were Africanized bees in every county of the state, and they stopped doing this quarantine because they knew that it was useless to try to contain bees. They would move from, because the swarms move readily between trees and locations, they couldn't contain them. And so the funding for this program ended. So if you look up spread of Africanized honeybees by year anywhere online, this is the latest map available, which is 10 years old now because there was no funding anymore. And it is assumed that in most southern US states, there are Africanized bees. Interestingly, there are not reports of Africanized bees in some states, including Mississippi, for example, but I suspect that it's because people haven't tested them, not that they don't exist there. So the introduction of honeybees, Africanized lineage bees into the Americas was a double-edged sword. It was a huge success in many ways, but it also brought new issues. It displaced the native bees at the very beginning because they're very aggressive in trying to get the resources. It almost eliminated hobby beekeeping because it was just, and it continues to be kind of annoying to work with these bees. It's not as simple anymore. There was definitely loss of livestock and humans due to stinging deaths. There was little initial profit to be made because the operating costs went up. Africanized honeybees abscond and swarm at a very high rate. So sometimes you lose the colonies that you want to keep in your boxes for high or large population and thus higher honey productivity. And typically with Africanized honeybees, the resources that they collect kind of are kept by them. So they don't really hoard honey nor pollen because where they live, they don't have a true winter that they have to save up honey for. They typically have mild winters, and that's why Texas is such a suitable climate for them. And so their blooming season for plants is not totally halted for 4 or 6 months of the year. It's more like 1 or 2 months, and then we have something that could try to keep them going, either in terms of protein or carbohydrates. Of course, the most infamous trait of Africanized honeybees is their defensive behavior or their likelihood to sting. And so research has found that this defensive behavior is genetically dominant or additive. Several genes are involved in this likelihood of being more aggressive. It's not just one gene. And as you may all know, there are Africanized bees that are very gentle in demeanor, and there are European colonies that are very aggressive in demeanor and everything in between. So being Africanized does not tell you right away that a colony is supposed to be incredibly aggressive and vice versa. But in general, Africanized honeybees have a lower threshold for defensive behavior, which means that it takes less stimuli for these bees to start an attack and or releasing alarm pheromone. They're 4 to 10 times more likely to escalate to stinging behaviors. You know that stinging leads to death, so they will often, European bees will undergo behaviors that are kind of pre-stinging, kind of showing tougher than you kind of behaviors that are trying to win a combat without engaging in stinging because the bees die after stinging. But they're more likely to escalate to stinging if they're Africanized. Their alarm pheromone recruits 10 to 30 more bees, 30 times more bees, because they release more of this alarm pheromone and because they have a lower threshold for activating their own behavior when they smell the alarm pheromone. And therefore, they also pursue for longer distances. So you can have Africanized bees pursue you when you bother them for up to 1,000 meters, which is really not seen typically with European honeybees. After 100 meters or so, you typically are okay after disturbing European bees that are kind of hot. But again, size and defensive behavior are not genetically linked. So now all of the tests that are done have to be done with genetics and with molecular tools, because no one is now convinced that you can readily detect Africanized honeybees just by sight, because there's such a range in sight. There's small European bees and there's large Africanized honeybees. And so therefore they mix in within that range of what should be the appropriate size for each type of bee. So the initial impact on the beekeeping community for these, after the introduction of Africanized honeybees, is that apiaries had to be isolated from people and livestock. That continues to be the case in many regions. Apiaries had to be smaller and more spread out to prevent explosions of high density colonies right next to each other, and then kind of priming each other for very aggressive attacks. In regions like South America and Central America, people use stronger smokers. Even here, you buy the bigger smoker compared to the small smoker, and definitely the PPE, or protective protection equipment, is not it's not just recommended, it is required when you're working with these bees. So typically people work in teams when one person is the smoker and the other one is dealing with bees, so that there's smoke constantly being applied to the colony to tame them. Initially, some beekeepers went out of business. Beekeeping became more expensive. In Brazil, beekeeping dropped to 10% of its former values, at least at the beginning. And because a lot of people dropped out of the race, honey production actually went down by 3,000 tons per year, or 40%. But after 10 years, honey production bounced back, and now it's over 50,000 tons per year. So a lot of the assumed benefits of the introduction of the Africanized honeybees into the Americas, or at least in Brazil, was realized later on, and people have learned to deal with them, including our own state of Texas. So even though there are no surveys conducted anymore by the USDA or APHIS, a few papers have done some work on the predictive expansion of Africanized honeybees in the US. And so this particular paper is not very new, from 2006, but it had correlated, which I like this paper because it was in 2006 and he was already talking about climate change and rising temperatures, average temperatures. And so it said, they predicted that this area of the light gray in the United States are areas that within about 20 more years or so after this study was done, up to 1980, these are areas that are predicted to have 120 days or more that have nice enough temperatures for the Africanized honeybee to at least conceivably be okay to thrive in those conditions. In the area where the white is, it is too cold, and temperatures will be at least 120 days in the year, the temperatures will be below 10 degrees Celsius, which is too low for Africanized bees. So this was in the dark gray, this was the range of the Africanized honeybee in 2006. Now you know that it's a little more spread out in other parts of the southern US. But this light gray is the predicted range in which bees would potentially be able to thrive, if we consider that bees are not able to thrive under 10 degrees Celsius for more than 120 days out of the year. This one is one of the more up-to-date studies by Stoker and et al. And what they did was, they did all of these mathematical analyses of weather data, again, taking into consideration basically climate change. And they came up with predictive regions where Africanized bees and European bees would thrive in the US in the next 20 years or so. And so in the Western Great Basin, the Great Plains, and Southern Appalachian Mountains, these regions in red and orange are what are considered by the authors to be zones of tension, where models show that European honeybees may be struggling in the future to maintain their dominance in the US. In the yellow means basically that sooner or later, because of higher temperatures and suitable locations, the Africanized bee might live all the way up to the Pacific Northwest. the European bees will maintain their stronghold in this part of the country, but in these regions of the orange and yellow and red, those are the zones of tension. So really cool studies. So now that you know kind of where they came from and where they might be going here in the US, now I want to tell you about the studies that we've done with these bees here in Texas. So first, I want to tell you about the precise Africanization patterns in a population of feral honeybees in South Texas. So the interesting thing about this is that we're dealing with feral or wild bees. And by wild, we mean living in wild conditions and that have been unmanaged for many, many years, and they live by themselves. So we published this a few years ago. So, but before us, we basically inherited that system from people that came before. They did an 11-year survey in 1999 to 2001 of a population in southern Texas, and they looked at the mitochondrial DNA of the bees in those colonies, meaning that this type of DNA is only maternally inherited. So it's all through the matrilineal lines that you can detect the lineage descent of those bees. And so they went on to, and I will show you pictures of this location shortly. They went to this location called the Welder Wildlife Refuge in South Texas. It's near Sinton. And they surveyed bees in this region, in this one refuge from, again, 1991 to 2001. For the first 4 years, they barely saw any bees that came from Apis mellifera scutellata maternal lineage, which is the South African lineage that we're interested in. There were some bees of Eastern European descent, some bees of Western European descent, and this one, which is of the old lineage Lamarckii, which is kind of the Egyptian honeybee. But then they started seeing maybe 1 or 2 colonies that had now Africanized lineage. Then you see a complete drop within a year in the number of bees of European descent and an increase in the number of bees of Africanized descent. The mitochondrial, as I repeat, a mitochondrial DNA, so maternal lineage went up in the scutellata lineage for these colonies. So this location is called, again, the Welder Wildlife Refuge. It's 60 kilometers north of Corpus Christi. So here's Corpus, here's Sinton, and here's the Welder Wildlife Refuge. And I encourage you to look them up because they are a cattle ranching, petroleum, gas exploiting reserve that the owners wanted a place. It's called Rob and Bessie Welder. They left an estate where they can make enough profits from gas exploitation and sustainable cattle ranching that they can also help in doing conservation and wildlife maintenance of the rest of the wildlife in the refuge. So it's kind of, if you read about them, they will tell you about how they have combined both their love for conservation and an ability to financially maintain the foundation and the refuge through gas revenues, and they actually fund a few graduate research fellowships every year. So it's 7,800 acres, 80% covered in chaparral, brushland, live oak, moss, very humid. As if you've been there, it's really, really hot and humid in the summer. So this is an aerial picture of the welder used for GIS, or a geographic information system analysis. And these little dots are all the locations of trees that my colleagues before me and then later us mapped on the Welder Wildlife Refuge. So a tree looking like this might harbor a colony of bees. So they did the first collections in the first decade. Then we went back in 2013. We went back in 2016, and we just came back this past August. We went back to the welder. I'm not going to show you data from this year because we just collected it. So 85% of the bees in these and the trees that we found were living in post oaks. In 2001, they found 112 bee trees. We actually found fewer. So the bees that they had luckily saved for us all these years, we collected a couple of bees from them for a couple of studies. So here's the first type of information. This one shows the annual distribution of the cavities between 1991 and 2016. And so here, this bar graph shows you something similar to what you saw before. Sorry, this is just occupancy. So the number of bee trees that we found per year, so in this case, let's say like 90 bee trees, and the percentage that was occupied. So that percentage is shown in the red dots. So in 91, 100% here is the other y-axis. For example, in 1996, only about 41% of, or less than 50% of the trees that we found were occupied, etc. So we surveyed 6.25 kilometers square in the first survey, only 5 kilometers square in the later surveys. And in the last survey in 2013, we only found 28 colonies out of 89 trees that were surveyed, because the rest of the other trees actually died. There's a lot of rotting going on of trees definitely near the ravines and rivers, very humid, and you lose a lot of trees. They die year after year. And 32 of 97 trees were occupied in 2016. So 33% occupancy or so. This is the one that looks very similar to the one I showed you in black and white, with these bar graphs showing all of those lineages that I showed you earlier. But what's cool about this one is that it's now up to date with 2013 data. So 25 colonies out of the 28 were Africanized, so 90% of them. 3 colonies were of Western European maternal ancestry, which is in the blue. And you actually we lost Eastern European bees and the Lamarcki bees that they had found in the previous survey. So that's unfortunate, because they lost basically diversity of bees. And so these dots are just showing the percent of colonies that were of Africanized descent. And in this case, it's 89%, sorry. Now, if we just finish this off with 2016 data, an interesting flip happened. We found no bees of Western European descent, but we found bees of O lineage, but only really 2 colonies, and they were both of O lineage. So somehow bees are getting into the refuge from other locations, or what might be happening more commonly is that we didn't sample enough bees around, and we were missing some of the O lineage bees that were in the vicinity of the survey trees. So only 6% of colonies were not Africanized in 2016. I'm really curious to know what the numbers are for what we got in 2021. Okay, so for the genomic DNA analysis, so we did the analysis that I just showed you for the mitochondrial DNA. The mitochondria is an organelle within the cell, and it's all the DNA contained in the mitochondria is acquired from the mother, not the father. Whereas the rest, the cell also has a nucleus, and inside that nucleus there's DNA. We call it genomic DNA or nuclear DNA. That one comes from both the father and the mother. It's not just maternally inherited. It comes in equal proportions from dad and mom. And so with that type of genomic DNA, you can do a similar but different analysis to look at where your bees basically come from. So to look at genomic DNA, you have to have first a sample of DNA that is genomic DNA that is fully European. So it's brought from bees that came from Europe. And you know exactly that those bees are fully European. And you also have to have DNA from bees that are fully of Apis mellifera scutellata descent at the genome level from both mom and dad. So you have to have the true or almost pure individuals to compare your samples to. And so here's the analysis of the genomic DNA from 1991 all the way to 2013, taking into account that European bee genome looks green on this graph and Africanized genome looks of African descent looks red. And so as you can see, you have less and less green over time. And then by 2013, we had very little importation or maintenance of that genomic DNA from bees of European descent in that population at the Wildlife Refuge. So we have this increase of Apis mellifera scutellata derived genes at the genomic level in this population. So again, we examined gene flow patterns in this population 23 years after the first hybridization event occurred. We found only about 33% occupancy. So in 2013, again, about only 30% occupancy. And of these, 90% were Africanized, no Eastern European colonies in those samples in 2013. We also examined that gene flow in the genomic DNA 26 years after the first hybridization event occurred. 32 colonies were occupied, trees were occupied in 2016. Of these, 2 colonies were of O lineage and the rest were Africanized. We did not find any bees of Eastern or Western European maternal ancestry. That genomic DNA analysis revealed a high integration of Apis mellifera scutellata derived genes, suggesting that the feral population is an admixed swarm with a now greater presence of African derived both nuclear and mitochondrial genetic composition. So this is the work that I'll talk at the end. The ongoing work is the assessment of pathogen loads in this population. And we're working with people at Penn State to see if there's any type of parent of origin gene expression between crosses of Africanizing European queens and drones to see whether it is more likely that a Africanized mother will imprint their genes onto the offspring compared to a European father, for example, to see if fathers or mothers are more prone to passing on their genes and gene expression. So we're looking at retinue behavior, ovary activation behavior, aggression behavior in those assays. Let me look at the chat, see if there's questions here. Oh yeah, it was the reference that, yeah, that was a long time ago. Yes, that's in Brazil. From what amount, I don't know the amount. I can inquire for you, but it went down by 3,000 tons in Brazil after the first few years. So now I'm going to talk about the prevalence of nosema in this population of feral bees. And this basically was a 20-year study that took advantage of those old samples that had been collected in the 90s. So Microsporidia is a group of parasitic fungi that has over 200 species of nosema. And nosema causes nosemosis, which is one of the more prevalent diseases that affect honeybee adults. And in the US, it's caused by 2 species, Nosema apis, which was the original pathogen of Apis mellifera, and now Nosema cerana, which was the original pathogen of only Apis cerana, the Asian honeybee. And in the last 30 years, we saw a switching or jumping of hosts from Apis cerana to Apis mellifera, and now Nosema cerana infects both species, and it's actually thought to be more virulent or cause more severe disease in many areas. So infection begins when a honeybee consumes the spores, typically when they're cleaning out spore-infested feces from comb, and then they share it with each other, and then within a matter of a few days, the bee can have millions of spores in their epithelial cells in their midgut. So colonies infected with one or both species have reductions in worker population and honey production, typically caused by early mortality of the worker force, mostly because of the digestive disorders that Nosema causes. And so you've seen streaking in the hives of Nosema apis infected colonies because you don't see the fecal streaking in Nosema cerana infected colonies. So basically bees get diarrhea with Nosema apis. They can't really go and void themselves outside of the colony, which increases the contamination of the colony. The spores of the two species are very similar in shape, and thus they're very difficult to differentiate among each other. So you can see in this picture, Nosema apis tends to be a little more roundish. They're both oblong, but the one is a little more, has a bigger belly, and Nosema cerana is a little slimmer. But as an expert wrote on this slide, they are not sure which one you would consider this one because it kind of seems to be in the middle. So it's very difficult to rely on microscopic assessment of Nosema. Nowadays, these are done with molecular tools. Nosema apis was believed to be the only species to infect European bees until 1996, when cerana was found in managed colonies, and now it is present in all continents, but no one has really looked at Nosema in areas where Africanized bees thrive, and most importantly, where feral Africanized bees thrive, because very few studies that I looked at Nosema in Africanized honeybees have been done on managed colonies, including a couple of our own that I won't talk about. So we wanted to see what the levels of Nosema were for these colonies over the last 30 years, using the same population, the same sampling technique. Because we are not allowed to intrude in these preserved colonies in bee trees, we have only an ability to sweep bees using a net outside of the colony. So we're only, unfortunately, we can only sample adults that are right outside foraging or guarding. So we estimated the average number of Nosema species per bee. That's under the scope, but we can't tell the species. Then we did work with a collaborator at Penn State, Schulhill, where we did the DNA work to determine the actual species. I will bore you with the details. It's called quantitative polymerase chain reaction, or qPCR, to determine what is known as DNA copy numbers. And so if a specific species is present in your sample, then you should have some DNA found in that sample, and it'll tell you kind of the relative abundance of that specific pathogen, for example, through DNA copy numbers. And you will see something similar in a couple in this study and the next one that I'm going to close with. So we sampled about 10 bees per colony, and we only analyzed a subsample of our years because this is expensive work. It's really expensive to do molecular work. So we analyzed a total of 474 workers, of which only 28 tested positive for Nosema spores under the scope. So that already tells you that there's very low infection rates. And those that were positive had a very small number of loads. Because if you think about, so 10 to the 3 is 1,000, 10 to the 4 is 10,000, 10 to the 6 is 1,000,000. Typically, a high infection rate would be between 4 and 10 million spores per bee, but these bees are only infected with 1,000 spores, or in this case, 13,000 spores. So nothing in the millions like managed bees have. So this one shows the levels of Nosema apis. And so the Nosema apis was present in about 30+% of the samples in 1991. These numbers represent the number of colonies that were sampled. And after 1995, you don't see any bees infected with Nosema apis. The opposite was the case with Nosema serrana. Nosema serrana was found in all the years that we have sampled bees, including 2013, which was the last study for Nosema. 85% of the colonies were infected in 2013. And so when you combine the results, this now is not just number of spores and percent of colonies infected. This one is the one that does the quantitative PCR and the DNA copy number. So this one tells you the copy number. You see both species in 91, in 92, in 94, in 95. You don't see any in 96. And then that's it. Bye-bye. There's no Nosema apis ever again. And then the numbers of Nosema serrana have gone up to above levels of concern, which are like in the tens of millions per bee in 2013. So it is more infectious than Nosema apis for sure. So the level of Nosema in these feral bees have changed over the last 20 plus years with interesting population dynamics. The number of colonies with Nosema serrana increased from 18% in 91 to 86% in 2013, while the percentage of colonies of Nosema apis dropped from 33% in 91 to disappearing since 1995. So this follows the pattern that has been seen in other countries and in other places in the US, that Nosema serrana has displaced Nosema apis in this population of feral bees. And so there's something really interesting happening between the two species where one is outcompeting the other. But we definitely need to continue doing more work, and that's what we're doing right now. So I'm gonna finish off real quick with this study that is it's basically the whole PhD dissertation of my student, Myra Dickey, which, you know, in the future, I invite you to invite for a presentation. She's great. She's a third-year PhD student. And she wanted to combine her passion for molecular, tools in the laboratory, beekeeping. She started her own beekeeping club at Salisbury University, where she did her undergrad in Virginia, and her interest in Africanized bees. So she's now taking on these projects with Africanized bees at the Wilder Wildlife Refuge, and she's looking at pathogens in that population. So we know that these colonies are found in tree cavities, and they have smaller colonies, and those are the ones that we are looking at. So what she found looking for whatever is out there in terms of wild versus managed colonies and epidemiology is that wild and managed colonies, for many reasons, have different evolutionary pressures. So from these papers in 2016 and 2017, because you have low colony density in wild areas where they're unmanaged and they live in bee trees, in tree cavities, they're very spread out, and therefore there's very few pathogens, and also there's very low likelihood that the bees are going to drift and infect other colonies. Whereas when you have high colony abundance, like in this diagram, you have more infected individuals and a higher likelihood of drifting and infecting each other. So parasite loads are positively correlated with colony density at the landscape scale. And populations not treated against parasites will evolve resistant mechanisms through natural selection. And that's what we're thinking, that these bees in wild settings are probably survivor bees that have gone through all of these bottleneck effects from disease incidents. So typically these wild colonies have low pathogen and parasite loads than managed colonies. The ones that had really high levels are probably, they die out because they get too sick, they're untreated, they die out. So the ones, the survivor stocks, typically maintain low pathogen and parasite loads through many mechanisms, including, for example, behavioral traits like varroa-sensitive hygiene behavior or hygienic behavior in general. So no one really has looked at viruses in either African bees in Africa or Africanized bees in Africanized settings. So they've done a study in Kenya where they found all of these bees, all of these bee-associated viruses in Kenya, in some areas in Africa. And with Africanized bees, not in Africa, but in the US, all of the studies have been done with managed bees, not wild bees. And those have found all of these, Israeli acute paralysis virus, black queen cell virus, deformed wing virus, acute bee paralysis virus, Kashmir bee virus. So we wanted to think, or we still want to know whether the prevalence of viruses is lower in this unmanaged population of Africanized bees in southern Texas. So the objective of her whole PhD, well, part of her PhD, is to identify and quantify honeybee-associated viruses in this population and compare it to the presence of these viruses in the nearest managed apiary to see if there are differences in the levels of viruses and types of viruses between managed and unmanaged bees. And we think that we will find higher levels of viruses in the managed populations because of all the reasons I just stated compared to your unmanaged wild colonies. And so here's Myra collecting bees. We use nets. As you can see, that colony is actually not very aggressive. It actually, for many of these, it took several times us pounding the entrance. You see she's right at the entrance and the bees are not even caring that we're sampling them. Some of them were aggressive, but not unmanageable. So we collected bees again this year in 2021, and if we get funding, we'll go back in 2022. And we use the ones from the previous samplings, and we extract RNA because all of these viruses are RNA viruses. They're not DNA viruses, they're RNA viruses. So it's basically the one strand instead of double-stranded DNA. And we screen these samples for 9 different viruses. All the ones I just told you, but also Lexini virus. I don't remember what VDV stands for. Sac brood, the other one that I hadn't mentioned. So we do this through quantitative PCR that I told you about, and we do the same thing that we did with Nozema with genome copy number. And we compare, we will in the future, we haven't done this, compare the levels of viruses in these bees to the managed apiary. So Myra collected samples from about 40 colonies for the closest beekeeper to that area, which is 30 miles from the welder, though no one else manages bees nearby. So most of the bees in that area are thought to be unmanaged. The owner of the wildlife, well, the director of the wildlife refuge has 2 colonies. So we have a sample of 2 on site that we'll also look at. So these are the preliminary data. Really interesting results. Here on the right, you have the levels of viruses that are considered high for that specific virus, like Lexini, the deformed wing virus, black queen cell virus. So anything over 1 million copy numbers or more is considered high for Lexini viruses. For the deformed wing, it has to be in the order of 100 copy numbers or 10, anyway, and then 10 to the 8 or higher. So we had 6 samples where we had enough bees collected for this type of analysis in 2013. Of the 6, we didn't detect any viruses in 2 samples, and the rest tested positive for Lexini, black queen cell, and the deformed wing. Here are the results. As you can see, the copy numbers are really low, definitely below what is considered a high viral titer for any of these viruses. And here are the results for 2016. We sampled 20 colonies. We didn't detect viruses in 7 of them, and the rest tested positive for 3 viruses, sac brood, deformed wing, and black queen cell virus. And these are what are considered high titers. None of them went even up to the 1 times e to the 6 or 1 million copy numbers per individual. So we find these viruses that are really low level. So now we have, what we're doing is, well, and then we also wanted to compare the levels across years to see if there have been differences across years. And there's no difference in, at least in the foreign virus, between 2013 and 2016. We'll see what happens with the levels in 2021. So all of these estimates are considered low, which could explain how these wild colonies have been able to survive at this Wilder Wildlife Refuge and many places probably around Texas where we have kind of, forests and parks in the absence of human intervention. So now we're analyzing all the data that we collected this year and hopefully in 2022. So I hope to have given you a mouthful of information about Africanized bees, their ecology, how they spread in the Americas, and how they're doing, at least in the Wilder Wildlife Refuge here in Texas. So I want to thank our collaborators. I highly encourage you to, if you don't follow us yet, to follow us on Facebook. We have almost 5,000 followers now. We actually now have Instagram and we're trending. I don't remember what the handle is, but basically you just look up the same thing, Tamio Honeybee Lab on Instagram. This is an old thing. This is now like over 4,800 people following us. And so that's it.
All right, Dr. Rangel, we do appreciate it. It's great hearing from that. Has there been any thought of, I know y'all are down in that lower area, but like testing up in towards our area to see, if there is a population of the Africanized honeybees in this area?
Oh, I guarantee that your bees, anything that's in meter boxes and sheds and are smaller in size are probably Africanized. So our lab can test for Africanization. We charge, because it's expensive. What we recommend is that basically you assume that they are Africanized based on their behavior, and then immediately requeen if possible, because except for satiating your curiosity, knowing that they're Africanized, it's not going to do much for you. Basically, you're going to convince yourself that you needed to requeen anyway. So it's expensive, but if that's what you want, there are laboratories that will test for Africanization.
Sounds like they're doing better if you can get them Africanized.
That's true, except they're hard to manage in some cases. But if you could Google GAHB or gentle Africanized honeybee, and then the country Puerto Rico. They have a very interesting population that they keep studying because it's just really gentle. And somehow they're moved from, just because of these bottleneck effects that occur when they invade small countries or small islands, only a certain population of these bees made it to Puerto Rico, and some of them were of really gentle demeanor, and then they probably bred them more and more, and now they're just totally gentle and they're Africanized. Whereas I've studied bees in many countries in Central America and the Caribbean, and I have bet several times that the bees are Africanized and they're not. So that's two things. That brings me back to two things. One, they're not Africanized at the mitochondrial level, which is the usual test that people do, but we didn't really test them for the genomic DNA. If we do that, where you do that, you compare it to the European and African, the green and the red, then you're probably going to see that a lot of genes come from African descent, and that might help explain why they're so aggressive. and so that's another point. And the last point I want to make is that if I bet then, if that's the case, that if you did that study with the green and red reference populations of genomic DNA, you will find in northern states of the US, you might find some red in that type of analysis. So the key question that no one can answer yet is what percentage is needed for these bees to show the aggressive behavior of Africanized bees if they only have a little bit of genomic DNA of African descent? That hasn't been answered to my knowledge, but it will probably be very interesting to continue to work with Africanized bees because the interest kind of stopped for a while. There's some revived interest in these, especially in these unmanaged populations. And because no one has, they kind of stopped, or most people stopped looking at them for like 10 years or so in the US anyway. They might look at them in the Americas, but I don't know of any study that has looked at wild bees in those settings. They typically just study their apiaries, and most of their apiaries are Africanized anyway, so that's what they look for.
It is neat to see what kind of percent we're getting up here in the northern Texas area, versus that. Are y'all still doing any lab work down in the Westlaco area, or
No, that laboratory shut down about over 10 years ago, and they were no longer, or if there were any bee scientists left, they moved to other USDA labs. So there's no bee lab in Texas. The closest is the one in Baton Rouge, Louisiana, which is actually very active now. They have wonderful people working there, a lot of bee scientists in that lab. And now, interestingly, I don't know if any of you ever saw my former student Pierre Lau speak, wonderful speaker. He's now a USDA scientist in Mississippi. And for whatever reason, there's now a group of like 8 bee scientists in that same general area in Mississippi working with either the USDA or the University of Mississippi or Mississippi State. So there's kind of a nice bee working group in Mississippi that potentially at some point might start studying Africanized bees, because I think it's just a matter of time in that state that they would become a problem.
Yeah, I hate losing Pierre, but he did a lot of good stuff down in the area.
That's right. Yeah.
It looks like you had a couple of questions on the chat.
So again, the percentage of genetic makeup that qualifies, we don't know. There's so many variables, and there's what we call epigenetics. So there's several genes that are involved in one trait. So it's almost impossible to tease them apart. The genes that call for one behavior are several, and so we don't know the magic number for that. I don't know if we ever will, but we definitely don't know yet. Are the colonies in Brazil that have become calm in a laboratory environment, or are these bees kept by everyday farmers? So in the laboratory, they definitely have basically bred for gentleness. In our lab, we kind of do the same. We do have a few Africanized colonies because we do work with Africanized bees in our lab, and the ones that are too aggressive to work with, we have to basically kill the queen and requeen. In countries where people are knowledgeable enough, basically your sideliner and commercial beekeepers whose livelihood depend on keeping bees, they have undergone breeding programs that work on, increasing the gentleness of these colonies so that they can work with them. Very different from very small scale beekeepers, farmers that are not very wealthy, that can barely, make ends meet, and they use beekeeping as a source of income on the side. They don't really do any training. I've dealt with a lot of them. They don't do a lot of training. They don't have the tools to get trained or the people to train them. They don't really do any breeding. And so their bees are very hot, very, very hot. And all they do is manage other things. So they know that their apiary can only be very secluded. They know that their neighbors know that they have bees. They go in and put on their suits before they even get to the apiary in their car. They know that they have to never take off their suits until after they've left the property and they're a few miles away. They know that they can't keep livestock next to the bees. So things like that, they know that they have very aggressive bees and they don't do much because they can't afford, they don't even know this whole idea of breeding doesn't really exist in their mind. So it depends on your level of education as a beekeeper and your resources, and that's really, really variable.
All right. Any more questions? Pop them on the chat real quick, and then.
Yeah, how do they even get back in their vehicle? So you just you do not take your suit off, and your cabin is full of bees. You shut the door so you don't get shut the window so you don't get more bees in, but your cabin will be full of bees, and then a mile away, then you lower the window or you start lowering the window, they start escaping until they're gone. But that's all you can do. Only until there's no more bees can you take off your suit, kind of a mile down the road. It says that your question above was not answered.
Yeah, he's up above. It's about the, it seems like bees.
Oh, there's a statistic about having only 25% of feral population survival in the first year. That's true. That's true. However, this statistic, which was actually done by my PhD advisor, Tom Seeley, it's actually 1 in 5 for lowers, 20% chance of surviving for first year swarms. So you're right. It's, well, two things. First, that statistic is was done with European bees in upstate New York, totally different environment, and European bees. No one has done the same for Africanized bees, feral Africanized honeybees. I wish I had the money to send someone to the welder more than once a year so that they could track survivorship of first year swarms. We don't have the capability to do that. That would be beautiful to do because no one knows. But second, when you talk about survivorship in this population, what we detect as a colony in a bee tree, we don't know if that's a first year swarm or has gone past its first year. Once a colony survives its first year, once a swarm moves into a bee tree, it only, as he said correctly, only about 1 in 5 survives. But the one that does has an 80% chance of survivorship year after year. So depending on when you do the snapshot of looking at a bee in a colony in a tree, you may be looking at probably something that survived its first winter, especially if we're sampling, maybe I did not make this clear, we sampled in June, July, and August. So potentially those colonies already survived their first year. And so we're looking at something that will survive probably, likely, is more likely to survive to the next year. But you're right, we cannot make any assumption that, let's say, because this time around we were able to sample bees in 2021 and 2022, well, hopefully next year, 2 consecutive years. If we find bees on the same tree cavity, we cannot say with certainty that it's exactly the same colony because we were unable to check that colony then mid-year to see that it's still there. I mean, it's very likely if we mitotype it and we find that they're both O lineage or a specific line of A lineage, that it might be the same colony, but we won't know for sure with 100% certainty, because they could have, by random luck, maybe the first colony in the first year died, and then a new swarm moved in that had the same lineage.
And were they affected as much with this freeze that we had?
Oh yeah, so that's the thing, that this year our colony density is much lower. I think we found only bees in about 20 trees, compared to 30, 33 the last couple of times. And Terry, the director of the Welder, thinks that it was definitely due to the freeze in February. If you saw some of those pictures, they have really wide openings. And it's because these bees don't really undergo very terrible winters. And so they're probably okay with a little bit of drift, in the winter, but nothing like what happened in February. And he said that it snowed out there, and so it's basically probably killed a lot of the bees that would have, that maybe have survived through February, and then February hit and killed a lot of the bees. So unfortunately, we didn't go there last year, so I don't know. It would have been really nice to do the assessment of what there was a year ago compared to this spring after the freeze.
Okay. It looks like pretty much all the questions now. If you have any last ones, you can always email her at her jrangel@ag.tamu.edu. I know my dad tried to bounce a few emails to you and had the old one, and it bounced them back. So
yeah, it's been a mess, because then we find a gazillion emails in the junk mailbox, and I never replied to them because I had no idea I had them. It's just frustrating, but it's supposed to get better with time. What do you call that, transition or whatever, it's giving us all a heartache.
I think it's happening
it'll
everywhere.
get better. Yeah.
All right. Thank you, Dr. Rangel.
No problem.
Folks, if you've not been down to the Texas Bee Lab, I suggest you get down there. Y'all don't call it Riverside Campus anymore, do you?
No more. It's called the Relis Campus.
Relic Campus.
Relis, because it's an acronym used by Aggies: respect, excellence, leadership, loyalty. I forget what the I is, and S is selfless service. Yeah.
Okay. But yeah, definitely get down to the Bee Lab, see what they're doing. The students are doing a whole lot. I know that the lab down there, it seems like it keeps growing, and y'all are doing more and more testing, and even got your own hives down there too. So
and we every year, well, besides COVID, and one year I was on maternity leave, but we do the art of queen rearing workshop. We don't have the dates for it yet for next year, but it always fills up, and we should be having it. I think it's either the last weekend in May or the first one or toward the end of May. It's going to be a half Friday, all day Saturday. And so if you follow us on Facebook, you'll hear about it and you'll know how to sign up. We're going to have Sue Kobe from University of Washington as a guest speaker. Hopefully, she said yes. Hopefully, she'll keep her word. Every time she says yes, she's come. And this year, we're going to have also Dr. Jennifer Shruda from the University of Tennessee as a guest speaker.
Well, folks, if you want to know about queen rearing, that's the time to go. Dr. Sue Kobe, great person there too for queen rearing. I think she's done, I don't know, I want to say millions of those classes.
Oh yeah.
So that's definitely one. But yes, Dr. Rangel, thank you so much. Well deserved on your John Thomas Award. And like I said, you've done a lot for the beekeeping community, and I know you've definitely helped us. And it was a great honor to be there when you got it. So
Thank you very much. Yeah, that was very touching, and yeah, it was humbling.
Yeah, we
Thank you.
appreciate everything y'all are doing. Matter of fact, I know that my dad we have the scholarship award. We were trying to give it to somebody locally, not
Yeah.
have anybody sign in, so we did shoot it through TIBA, and we
Yeah.
were wanting to make sure that it got over to one of the college students there. So
Yeah,
hopefully
wow,
we
that's
can
wonderful.
do that, and y'all will be able to get more funding to get these students to do more stuff.
Yeah, thank you. And maybe I'll see some of you folks at the TBA convention. We're gonna have our booth, our usual booth where we talk to people, come by and say hi. And I'm giving a couple of talks. We have Houston from the tech transfer team. He's gonna give a talk about the Be Informed partnership and what he's doing for Texas Beekeepers. And we have my student Taylor Reams, who works with Varroa and Varroa behavior. She's also giving a talk on her work on Varroa. Very interesting stuff.
I saw Megan's coming back too. So
Megan Mahoney, who used to be the Be Informed Partnership, the tech team leader, she was the first one. Now she's independent. Then we had Dan Orell, who is now a graduate student at Auburn University. And now we have Cade, and he's just great. He's a really friendly guy. And so I'm sure you guys are going to love him if you go and see him speak. And I'm going to give 2 talks. I think, I don't remember if similar to this one, and then the work that Pierre and our group have been doing on nutrition.
Okay, well, we're looking forward to it. Like I said, guys, if you haven't ever been to TBA before, definitely get there. There's a lot of information that comes out from that. And like I said, you'll hear the guys from the A&M lab down there doing their presentations also. Don't you usually have some of the students, besides the presenting there
Typically we have more students, but this year, because the Entomological Society of America, which is the national meeting for entomology that we go to every year, happens to be the weekend before the TBA convention. And so we're all getting back on November 3rd, which is a Wednesday. So it's just too much for the students to then go to Galveston, because they also are teaching assistants for classes, and they're already taking off like 3 working days. So it's just myself and Taylor this year.
Okay, well, we're looking forward to it. Thank you so much again, and we do appreciate the talk. Nice to hear that about Africanized bees and getting more updates on what's going on.
Yeah.
I guess we'll have to start saying we do have Africanized bees up here. So.
Oh, I suspect you do. Yes.
Well, thank you so much.
Thanks. Bye, guys.
Bye. All right, Collin County, this concludes our October meeting. Like I said, be on the watch on the newsletters. Like I said, we got our election process coming up. If you're interested in the nomination part, please get on to our business meeting next Monday. It is posted out how to get on. So be on there so we can get your nominations in there and the selection process there. So be safe and enjoy seeing y'all. Hopefully y'all be at TBA. And like I said, if you got a last few minutes and you want to help volunteer, the state fair is still available, and get in contact with my dad and we can fill you in a slot. So thank you very much, and good night.